Hello, everyone. My name is Yuhwa. It's my great pleasure to be here with you to give this presentation online. Uh, Let me start sharing my slides. What I would like to talk about today is artificial intelligence assisted single cell analysis for high throughput 3D imaging and its application for spatial biology. Let me disclose my conflict of interest. I'm a co-founder, scientific advisor, and consultant of NanoSelect Biomedical. I have financial interest in the company. As we all know through this meeting, that spatial biology is a further advance from the single cell technology. In the single cell, we are able to identify the genomic makeup, the genomic properties of each individual cell, as opposed to the ensemble average, that allow us to detect and study the characteristics of real cells and identify new and classified new cell types. As we move towards the spatial biology, we not only understand the individual property of the cell, we also can understand the spatial relations, the cell-cell interactions, put the cells in the context of the tissue or organ. So it becomes provide a lot more information than single cell technology. What we could contribute as a tool developer is to understand, to help the researchers to understand within each spatial area, what will be the makeup of the cells, not only from the genetics information point of view, but also to understand the phenotype, the spatial characteristics, the tomography of each individual cells. Imagine we can slice a whole tissue, a piece of tissue, into very small slides. And then we will suspend the cells in solution going through a flow cytometer system that is able to produce a 3D individual cell tomography. And that allow us to put together the spatial correlations within each of area. The enabling tool we're going to talk about is really image-guided cell sorter. We start with a sorter that is able to detect the two-dimensional image. In collaboration with NanoSelect Biomedical, my lab has developed a system. This is tabletop microfluidic image-based sorting compared to the regular fax system that shows only the single dot representing the expression level of certain fluorophores or proteins. Here, each dot, we could also construct the cell image in the two-dimensional fashion. So we have the spatial information associated with the downstream genomic information. Here, let me actually show you very quickly how the system is constructed. Instead of using any camera to increase the throughput of the system, since we have to be dealing with millions of the cells, we actually use a single pixel imaging techniques where each parameter or each color is detected by a photomultiplied tube. So what we detect is really the waveform of the cell passing through the optical interrogation area. And as a result, it produces a waveform. And then we use a technique of spatial temporal transformation to convert the temporal waveform into the spatial two-dimensional distribution. Okay. In order to accomplish that, we actually have to scan the laser beam as opposed to keep the beam as a static laser beam. And we use acoustic optical deflector that can scan the beam at about the 200 kilohertz. So every 5 microseconds, we scan the laser beam across the body of the cell. As the cell flows through the interrogation area within the microfluidic channel, we could record entire waveform produced by the two cell that allow us to reconstruct the two-dimensional image of the cell. Here is example how the system work. For example, if we put the drugs, dexamethasone, into the cell, and that can locate the protein to localize a protein. And here you will find that the drug treated the cells, the protein is localized near the nucleus area. Without translocation or without the drug treatment, the protein is distributed evenly over the entire cell body. Based on the area ratio, we could actually define a gate and then isolate the cells that is responsive to the particular drug from this spatial information. And experimental results shows that initially we have 50-50 mix with and without drug treatment. After sorting, we can obtain 100% purity, isolating 100% of cells that are responsive to the drug treatment based on the protein localization. We could also look into the cell cycles within the particular tissue area. Here shows a label-free cell imaging technique. Here we detect the fluorescence and also the nucleus scattering. And when in the cells in the G1S phase, and we can see a well-localized scattering spot. But when the cell enter the G2N phase, because disintegrations of nuclear membrane, and we will find the scattering pattern is actually almost distributed over a much wider area within the cell. Here is another example we could see tell without labeling leukemia cells versus white blood cells, and they certainly have very different spatial characteristics. Now we will move on to the three-dimensional cell imaging or cell tomography. Previously, we get the 2D imaging and we can isolate the cell based on the imaging features at the throughput about 1,000 cells per second. But we know that the two-dimensional imaging has limited information content. You will have much more information if we can create the cell tomography or three-dimensional cell image. To do that, what we try to design is a flow cytometer with the light sheet microscopy and confocal microscopy integrated over the flow cytometer platform. This is how it works. And maybe I should actually show you the next cartoon, and that will show the animation, how the system works characteristically. First of all, this blue part represents the light sheet, okay? This cube represents the area we would like to create the three-dimensional scanning or imaging. And the light sheet, laser light sheet, is scanning by the acoustic optic deflector. At every 5 microseconds, the laser being scanned through the cube once. Next, what we do is we will put this scanning light sheet microscope in the confocal configuration. That means we're going to put a pinhole here. However, instead of one pinhole, we are going to put an array of pinhole because the cell is traveling in the microfluidic channel at the speed of 20 centimeters per second. Here shows that, okay, as the cell travels through the first pinhole and the light sheet is scanning along this direction. And then through this pinhole, the PNT behind the pinhole is going to register a waveform. Okay, each slice of the waveform corresponds to the intensity or the signal produced by a specific voxel along this column. Now, the cell travel in the flow chamber, in the flow microfluidic channel, get to the second line, and then the second scan create actually a second signal. As the cell continue to travel and the scanning continues, what happens is after the cell passes the first pinhole here and enters the second pinhole, the cell has already been scanned in this example 10 times, and that create a waveform like this by the PMT. And this waveform can be transformed through the spatial temporal transformation into the image of this single slice. And now the cell has been flown to the second pinhole. And the same process will repeat as a scanning beam scans through the cell, as the flowing cell, and that will generate the second set of the signal corresponding to the image of the second cross-section. And this process will go on, and finally will cover the entire volume, the three-dimensional volume, and this is the entire signal pattern. Here shows a summary of what we just described. In 5 microseconds, okay, we actually create a signal for one single column, one single row. And then after 20 scans in about 100 microseconds, and we create a cross-section plan. And here show the first fluorescence, the second fluorescence, and the size scattering signals. And then after 1 millisecond, after the cell travels through 10 pinholes, and now we have all the information that can be transformed into the three-dimensional tomography for the fluorescence signal, for the second color fluorescence signal, and for the size scattering signal, showing the optical intensity or the protein density of the cell, including all the microorganelles. And here show the comparison of the 3D image. On the left is a 3D tomography generated by Leica SP8 confocal microscope. It takes 10 seconds to produce this image for one single cell. On the right, it basically shows the throughput of our system, okay? And in 1 second, it can take 1,000 cells 3D image. So we have a throughput improvement by a factor of 10,000 that allow the system to be very useful to produce spatial biology information that will need to handle millions of cells. And here shows one simple example that we could actually attach antibody labeled beads, antibody conjugated beads to the cells after some incubation. Some cells actually with 1 bead attached, some cells has 3 beads attached, some have 4, some have 6, some has as many as 8. And we create a histogram like this. And we can isolate any particular set based on the number of beads, as this example. We could also actually find that when we suspend the cells that actually are spatially connected in the tissue can remain connected. And then we could actually image both cells in this configuration. Of course, we could also use it to study T cell receptor activation and neuroantigen recognition. And here's another example to show how the tissue will respond, the cells in the tissue respond under radiation therapy, gamma ray radiation. And what we shown here, in the middle row is the gamma H2AX foci. Each, the red spot represent a broken double section DNA that is labeled by this fluorescent gamma H2AX. So this spot distribution represent the broken DNA by the radiation, or it's a representation of the degree of or extent of damage by radiation therapy. And you can see the distribution and the number of broken parts is very different. And here just showed an example that how we could actually reconstruct or basically literally tens of thousands or hundreds of the thousand cell image. Here we show a very small subset. Okay, again, this is a fluorescent image. This basically shows the spatial distributions of the broken DNA by radiation therapy. And this is a size scattering signal representing the granularity of the cell. And here shows that, how this information, the spatial 3D information, provide a lot richer insight than the traditional flow cytometer. Traditional flow cytometer allow us to detect the integrated intensity, which has no correlation with the foci count that represent the extent of the DNA damage. You can see the distribution is all over the place. We have high intensity, but low foci count, or low intensity with high foci count. With the tomography, we could actually numerically label every single spot and find all the spatial correlation. imagine this study can also be done with fish to understand the RNA or DNA configuration. Here is another example that we could actually study the label-free size scattering imaging. This is an entire histogram, but if we actually define the threshold aided by some machine learning algorithm, low scattering produce an imaging distribution that shows actually almost the cell body. If we pick the middle range and then we find there's some high scattering area for different cells. And of course, we can pick up the highest intensity area and you will find the focal point of the scattering from each cell. And here's another example to see how we can classify cell. Of course, in this case, we just mix HEK, HeLa cell, and MCF7 together. Typical microscope pictures can tell you HEK from HeLa cells, but probably it would be difficult to tell HeLa cells from MCF7 cells. With the aid of a computation neural network, convolutional neural network, we can help us to classify the cells. Again, with 2D CNN neural net, UNet, and with 2D imaging input. This is the confusion table. The confusion table improves if we actually use 2D CNN UNet with 3D imaging. Of course, the best result is obtained with a 3D UNet with a 3D imaging information. And here shows the corresponding t-SNE diagram. And similar study can also be done with the blood cells, and we have white blood cells. And again, we find the 3D CNN neural network coupled with a 3D imaging give us a best capability. Limited by time, I wouldn't actually continue. I would like to end my talk by acknowledging my graduate students and postdoctors, and also my sponsor from DARPA, NSF, and NIH, and also particularly my collaborator, my colleagues from NanoSelect. Thank you very much, and thank you for your attention. I'd be happy to answer your questions during the panel section. Thank you.